just so enormously privileged to know many of you. For those of you who I don't know, Luke's got my cell phone number and my email address. Just see him right after. So as some of you know, I'm a medical geneticist based here at Columbia University. Shout out to everyone who's made this happen. I won't go through all the names, but you've really, I think everyone in some way has contributed, and you've got homework to do. I'll warn you about this. I'll get to this in a second. But what I'm here to be able to do is to help enable you to be Dr. Mom, Dr. Dad, Dr. Grandma, Dr. Grandpa, whoever you are in the families here. Leah, who's going to be speaking right after me, is going to take this information or give this information to enable you to do that. And all of the information we're talking about today is absolutely available to everyone in the community. So if you feel you want to take notes, absolutely feel free. But we're going to have everything posted online so that you can be ambassadors and take this back to your families, to your doctors, your therapists, everyone who can be helpful. And the slogan that I have for you today is the care until the cure. So as we're doing this, what I mean by that is that we have things we have to do every single day to be able to take care and support our loved ones with KAND. On the other hand, we also want to be working towards better days. Days that we can be able to go beyond what we've learned to be able to do now, but we need to be able to do both of these simultaneously. As we're doing that, there is a huge amount of knowledge in this room as well as online for those of you who are joining us that way. And so what we need to use both this meeting and afterwards is to be able to collectively gather that wisdom, that knowledge, that experience, and boil it down to what I'll call best practices. So being able to figure out what's worked, what hasn't worked, and be able to take from that, distill from that, the things that will be helpful to the community in terms of supporting our individuals with KAND. As we think about that, the thing that I'm excited about is that there are a lot of new technologies in terms of new medical therapies being developed. And my job, along with many of the other scientists that you're going to hear from today and that are not even here today but are busy working at home, is to be able to take a lot of those new enabling technologies and apply them to KAND, apply them to KIF1A. And so I'm going to tell you a little bit about this. I will warn you in advance. I don't know what the time frame for this is going to be, but I can tell you that we work literally day and night, 7 days a week, in terms of being able to figure out what's going to work. Being able to make new friends, new partners in terms of going around the world to try and explore any technology that will work. And many of you have been very helpful in terms of opening some of those doors and making those connections. So I'm going to try and give you a very high-level view, very, very high level, of what you're going to be hearing at the conference. And I and my team members are here today and tomorrow and by email for as many tomorrows as we're going to have together to answer any of this. But I want to, at a very high level, be able to explain what's going on. So with all of this, I know some of you are very sophisticated and some of you are relatively newer in terms of joining the community, so I apologize for any of you who are beyond the 101 level. If you're at the 305 level, we can have the advanced course later this afternoon. But KIF1A fundamentally, and again, all of this is going to be available online, KIF1A fundamentally is what we call a molecular motor. So it's an engine in neurons, in brain cells, and in nerve cells throughout the body that's responsible for walking along these things that we think of as train tracks or microtubules. And that big blob up there is, we think of it as cargo, but being able to transport molecules through the brain cells to where they need to go so that those neurons can do their job, can function, can be able to help us think, to be able to help us laugh, to be able to move our arms, to be able to feel things, to be able to see things. Those are what all of those brain cells and neurons are doing. So you're going to hear from a lot of investigators today, but you're going to hear that sometimes this motor, this walking along the train tracks here, let me see if my pointer will work. For the folks that are represented here, almost all of us have changes within kind of these feet along those motors. There are problems where those feet then get stuck. They get stuck along those train tracks, and I'll get to it a little bit later, but part of what happens is when they can't then transport that cargo along those train tracks, those cells are not happy. They don't survive, they don't do their function very well. And it may be that this gets to be even more, I'm going to use the word toxic, but that these may actually stop or block the traffic from other trains from going. So it's not only that they don't get to their destination, but they may actually cause a traffic jam, and that traffic jam may be problematic. And so as we think about this, we're trying to think about strategies that will either take those trains that are derailed and get them off the train track so that other traffic can go through, or potentially ways of being able to get them unstuck and to move forward. And that's a very basic way that we think of that you're going to be hearing from today in terms of this. So some of you have heard me online talk about this. This is exactly the same thing that we talked about before. As we think about this, and you may be hearing about things when you read the news about gene therapy or being able to gene replacement or various things, the problem is not just that we need to replace the KIF1A that's there. It's not that this is a deficiency, but again, it's something that's stuck on the tracks, and fundamentally that's a different thing that we have to think about, is how to be able to get that train off the track and get a new train on the track. And so we're trying to figure out how to be able to do that, and we do have some really good ideas and ways of going forward. So I want you to think about that as well. As I think about this, to me, there are many, many steps for us to get to. And for you to realize, and I want to set expectations, for other rare diseases like this doesn't happen overnight, and it hasn't happened overnight. There have been remarkable advances, and we're absolutely going to beg, borrow, and steal from those advances from any other diseases that they've been done. We are not ashamed of any of this. But it doesn't happen overnight, and so I want to explain to you the foundation of what we need to do. As I think about doing this, time is not our friend, and so we try and do this many ways, many things simultaneously. Luckily, those of you who know me, I multitask a lot, so that's what my lab does. That's what all of us we have as scientists are doing. We have different groups multitasking and doing different things, complementing, doing them all simultaneously so that all of us can move this forward. So let me explain. I'm going to explain what we're doing, and I'm going to interject with the things that I need you all to help us do together. So number one is, as you've heard about already, is being able to just get folks diagnosed. Make sure that we know all the individuals out there that have KIF1A. This is not unimportant in the sense that, as a couple people mentioned earlier, the more common a condition is, it stands to reason more money gets put behind that. People in terms of government agencies put more money behind that. I absolutely know this is much more common than people appreciate, but we need to be able to get people diagnosed. I myself have written very powerful statements to saying what the standard of care should be in the United States for getting people diagnosed from a genetic point of view with exome sequencing. So this is very much out there for insurance companies to follow that guidance and those recommendations. But at times you are going to hear us ask you to testify, so to speak, in terms of how important that is. So that's the first one. The second one is what Leah's going to be talking about, and we call this natural history. Leah's going to explain what that means even more, but it means, again, taking all of our collective experience and understanding the diversity, because as you all have been talking with each other and know each other, you know that we're not all the same in the community. Eleonora has had some challenges. There are others in this room that have had different challenges, but we're all united by this range of challenges. And we need to understand that. And one point that I know some of you have been making, but let me just reemphasize this, that when we get eventually to number 8 over here, that's clinical trials, we need to have very good natural history data to know what this condition is like without treatment so that we can see the difference with treatment. And if we have really good, rock-solid natural history data that the FDA will take, there is a much greater likelihood that we don't have to randomize people to be on placebo or sugar pills, but that if we've got something that's working, then we can get, if not everyone, but more people on the medication, on the treatment that's going to be used. And this is proven, I've worked on other diseases, this has to be done at the beginning, the very beginning, and so that's why we've been bugging you guys in terms of asking all these questions, asking you to fill out surveys, and I will warn you that this does not end, right? So this is a long-term Every single year, we're coming back to you to understand, because for those of you that have children, your children are growing up, they're changing, they're developing, and we need to know how that's different for KAND or for KIF1A, okay? So those of you who are tired of us, I'm sorry, but it's important, and we're trying to minimize the burden for you all, but it's important. As we're doing this, we're trying to understand what are we going to say if we get to the point of a treatment, again, a clinical trial. What is the outcome we're going to look at to see a difference? Is it going to be differences like, for instance, fewer seizures? Is it going to be differences in terms of having the brain work better, have cognition better, being stronger, having less spasticity? So we need to think about that, and again, we need to have measures for that, and this is where your vote really matters. And so one of the things I'm in particular here listening about from you is, what is a meaningful difference to you? What makes differences in your lives? What would be most important as we rank order that? And can we actually find ways to measure that and measure small changes in whatever's meaningful? The reason I emphasize small changes is because we are going to want to know very quickly in a clinical trial what I call a go or no-go decision. So as soon as we can possibly get a signal that something's working, be able to double down in those directions, or as we're realizing that things are not working, I don't want to waste energy, effort, resources in going in the wrong direction. And so again, I need to hear very clearly from the community in terms of what's important, and then I will warn you again, we're going to be torturing you in the natural history study with ways of being able to measure that so that we can be very precise and very sensitive, be able to detect very small changes. One of the ways of doing that is actually not with something that we're going to see with our eyes in individuals with KAND, but something we might measure in the blood, something even potentially we might measure it in the fluid around the brain called the cerebrospinal fluid, but there are things that we call biomarkers which may give us sort of a signal of how healthy the brain cells are, how healthy the neurons are. And so I say this because many of you this weekend are going to be giving blood samples. Part of what you're giving those blood samples for is for us to be able to see at baseline for everyone today, what do those biomarkers look like? And so we're actually in the laboratory going to be furiously processing samples all weekend because we're measuring all of those different molecules in your blood. As parents, you're serving as controls. For your children, we're seeing what's different so that we can see, and I'll warn you again, next time we have a family meeting, we're coming back to you again so we can see if it's changed, if it's stable, if it's getting better or worse, but we need to actually have these measures over time. The other thing we're doing for many of you today, as you're giving samples, is we need to have a little bit of your child with KAND or KIF1A in the laboratory so that we can repeatedly try different things. We don't want to torture you. We don't want to have you come back from around the world. So for many of you, we're taking your blood cells, we're growing them in the laboratory. It's kind of mini-me in the lab. And that way, when we have either new drugs, new treatments, we can try them out. And I will say, and this is a really important message, many of you have heard me say, you're going to see a picture that shows all the different genetic changes in KAND and in KIF1A. And for many of you, you are the only one in the world that we know of with that particular change. Even for those of you where there might be more than one person or a few people, it's not very many. And you have to represent yourselves within those studies, because if you don't represent yourself, I can't come up with a treatment for your particular change. And I don't know how to say that any clearer. It is very, very important for you to represent yourselves, and I'll show you exactly why within one of the next slides. But we're all in this together. We're all pitching in, and I need everyone to do their part. As we do that, we make things, and I'll just throw out some of the language. We use things called induced pluripotential stem cells. We can make those into brain cells, again, in the dish. We can even make little mini brains, or what we call organoids, to be able to see how electricity travels around the brain. And as well, we have made and are continuing to make mouse models so that when we want to actually try something in a living organism, we don't have to do this first on your child or your family member. We can do it first on a little furry guy that looks very cute, but that is going to be able to try that for us. As we're doing this, we're still trying to make sure that I'm correct in what I said, which is that this is due to a problem with the train getting stuck on the train tracks. I won't go into a lot of the detail there, but we're still trying to make sure that's the case. And as we're doing this, we have a lot of companies, and I will be very clear. I'm not taking, I'm not have stock options or anything like that for any of these companies, so I don't have any conflicts of interest. But we are using anyone out there who has any science to bear on this, whether they're companies, whether they're academic researchers, whether there's someone who just had a great dream about this, I don't care. But as we're doing this, we're taking resources from anyone that might have it. So just to give you an example, there are companies that have gene therapy strategies. There are companies that have ways of being able to use what we call antisense oligonucleotides to be able to get rid of the trains that are stuck on the train tracks. But for any of those, we're willing to use any of those strategies. As we do this, and we have already started doing this for any of you that want to know, we have a huge mouse colony of KIF1A mice in the laboratory just a couple blocks over here. We have a huge number of them because we are doing little mouse clinical trials in the laboratory, just like we would do with people, but they're mice with KIF1A that are going through this and doing this. And ultimately, although we don't have it ready quite yet, we are doing this all in preparations and with discussions we've already had with the Food and Drug Administration. That's the agency in the United States that's responsible for new drugs and approving new drugs. So we know what they're going to require of us. We know that we could even do this with one person if we needed to. So we know what the path forward to be able to do that. And we do have also advice from them, given the diversity of the different changes we have in the community, about how to deal with some of those regulatory issues. So we have thought about, as I said, the entire sort of process that we need to go through in terms of doing this. I think we've got a good strategy, but it's hard. It's hard for the scientific community. This is not something that it's just taking an amoxicillin pill. I mean, I know you guys know this, but you've got a big team of people behind you in terms of doing this. So this is just basically a very similar thing in terms of what I was pointing out to, but I am going to include just a couple very specific points in this. Part of what we're doing, and you'll hear more about it from investigators today, is we are even building models in terms of being able to, for each individual mutation, that's what that means, each individual gene, be able to model that in the laboratory with literally molecular motors in terms of understanding that, and being able to get a better sense of what the prognosis is. I know many of you ask the question, what does the future hold? What can I expect? How quickly is this going to accelerate? And so we're doing those studies to be able to understand that. Some of you have given samples before, and those are in the laboratory working. I will say that although I, of course, never ever want it to happen to anyone in our community, we have had individuals that have given us enormous insight when their child has passed on to be able to help us understand what is going on. And so that's something that I can't thank the community enough in a time that's been really hard, what an enormous sacrifice they've made. We are going to have you give us more detailed information. So some of us have reported how things are going in terms of seizures or brain scans. It's really important for us to actually get the primary data. So not just to see a report of what an EEG was like or what an MRI was like, but we're going to be going through an enormous effort working with your doctors, with your hospitals, to actually get the primary EEG data. It's not easy, but we need you to point us and tell us who to go to get that, and getting the primary MRIs and being able to see differences over time, differences with different medications, with different treatments. This is now the major focus for the next year, is to be able to understand very specifically from you what medications, what treatments, what interventions are working, and to have the data to be able to see that and to understand exactly what a difference that makes. And so I will warn you, all of this is very scientifically driven, very evidence-based, but we need to be able to get the data and the evidence to do this. So those are the things that are in store for you, as I said, and you're going to hear from some of the investigators themselves in terms of the disease modeling. So we look forward to that later today in terms of doing that. We also have reached out to investigators at the National Institutes of Health, and so they have things like libraries of compounds, of small molecules or pills that could be used for this. They will help us in terms of the high throughput screening as we get there. And as I said, we're constantly talking almost once a week to new companies that have new strategies for gene therapies or for being able to get agents to the brain specifically. And so there are a lot of people behind the scenes helping. Okay, I'm going to try and explain kind of a complicated concept, but by doing it in the following way. As I was mentioning, as some people will be talking about later together, we literally have dozens of different mutations in KIF1A. And it is possible that we need to be able to grab onto one specific mutation and essentially make it dissolve, get rid of it. And it may be that at least one strategy would be to have a specific way of grabbing onto each specific mutation and then being able to make it dissolve. If it is true that we have to do that for every single different mutation, let's just pretend that we had 100 mutations, that would mean in parallel we would have to develop 100 drugs or 100 strategies. And that becomes not impossible, but challenging. It would be much better if we could boil that down instead of 100, for instance, to 10. That would make my life much easier, right? So how can we do that? Are there clever tricks that we can be able to do that? Oops, so here's what I'm representing. Let me see if I can go back. Okay, so each one of these X's, so there are 2 lines here. These are meant to represent the 2 chromosomes that an individual has that houses the KIF1A gene. This is meant to represent the X, the particular genetic change. And as you'll see, there are 1, 2, 3, 4, 5 people that are represented here. And there are 1, 2, 3, 4, 5 different genetic changes in KIF1A represented here because they're at different places along the gene. This is the length of the gene, and these are different positions here. What I'm trying to show you here is that all of us have other genetic changes in the KIF1A that are not bad, they're not problematic, they're not causing any problems, they're just normal genetic changes. And we may actually use that to our advantage. So let's take, for instance, this blue dot here. This blue dot happens to be on the same copy of the KIF1A gene as this mutation and also this mutation. That's great. Maybe instead of having to make a drug for this and this, maybe I could do a drug that targeted the blue dot, and then I get 2 for 1, right? Now I just cut my work down in half. That's fantastic. Can I keep doing that? Well, but let me be careful. Is that blue dot going to be tagging or traveling along with the KIF1A mutation in every person? And the answer is no. Right? So if I now look at this third case, here's the copy of the KIF1A gene, here's the problem right here, and here's my blue dot. But this blue dot for that person is actually on the good copy of their KIF1A. If I give that person medication for the blue dot, now I just got rid of the good gene. I don't want to do that, right? So this is a good strategy. I can come up and get a 2 for 1 for the KIF1A, but I only wanted to give it to number 1 and number 2. I do not want to give it to person number 3, right? So if you do that, and if you keep following back, let's take the purple dot as an example. So this person, let's take this one here. This person, I've got this purple dot here. I could use that to be able to tag my KIF1A mutation there, but now I go to this person here, and if I do that, and I use that to dissolve KIF1A, now I've dissolved everything, because they've got one on their one with their mutation, as well as one on the normal copy of the gene. Now I've got nothing left. Okay? So I have to be very careful how I tag this. So the reason we have signed many of you up to be able to get blood samples today, it's not just from your child with KIF1A, but it's actually for you as parents, moms and dads out there in terms of doing this. The reason we're doing this is because as we're doing this, it allows me to see one of these came from a mom, one of these came from a dad. And again, this particular genetic change per se did not come from you, but the rest of the KIF1A came from you. And I need to be able to see you as moms and dads are helping me to sort out which line this looks like, what travels together, what came from a mom versus what came from a dad. So as you're doing that, we're preparing ourselves with this entire inventory of the community about what these changes are so that we are ready to go as we have these, and we have these handles that we can latch onto. And again, it's for the purpose of trying to decrease the amount of work we've got to do and simplify things. Okay, so in the interest of time, I just want to wrap up by saying there's lots of things to do. We also, in terms of the natural history, are trying to, we don't have as much data as we'd like in terms of how our kiddos are growing. So you'll see we're going to be asking to measure things like head circumference, height, weight. It's an easy thing to do. Don't worry, I'm not going to embarrass anyone. We don't have to check weights moms out there, but we are going to check for your kids, be able to check and see how they're growing here at the meeting. Real simple to do. We're getting blood samples, and Luke's going to be coming back to you later. We have been working in terms of using the knowledge that we have to get additional funds out of the National Institutes of Health to support the community for doing this. And just stay tuned. We're close, but we're not quite there. So with that, I want to just stop, and I don't know that we have questions, time for questions now, or should we just go on?
I think we can do questions at the end, right before lunch.
Okay. Okay, very good. So next in line, I'm very pleased to be able to introduce Leah Boyle. I think many of you know already. So Leah is an MD PhD student here at Columbia. She is fantastically both smart and dedicated to everything KIF 1A. And she's going to be reviewing the natural history data that we have from the community in terms of how everyone's doing in the range of what we know. The one thing I want to emphasize that you'll hear from Leah is the diversity. There's quite a bit of diversity, and I don't either want anyone to panic about this, because if you see that some folks are having challenges, there is a diversity that very much depends on the genetic change, and so there are lots of differences between individuals because there are so many genetic differences. Leah?